Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll discuss with another topic, DABT topic, in vivo assay, induction phase of chemical sensitization. So let's directly dive into the question. The question is taken from American Board of Toxicology, Design, Executive and Interpret Toxicology Study Quiz, which was given by the DABT website in the website only, which is no longer available. There were a series of 50 questions were there. From that 50 questionnaire, this is one of the question. So what is the question? We'll see. This question is related to guideline, OECD guideline 442B, which is related to skin sensitization, local lymph node assay. So the question is, what in vivo assay measures only the induction phase of chemical sensitization? As we already know that sensitization have three phases will be there. Induction, then pre-sensitization, like the three phases will be there, no? So in that induction phase only, there measures only the induction phase of chemical sensitization. We need to identify the type of assay. So let's see what are the options we have. We have option number one, that is guinea pig maximization assay. Option number two, which is Drey's test, which is related to more like eye irritation and skin irritation test. Third option is mouse local lymph node assay, nothing but LLN assay. And fourth option is the Magnuson Kligman test. So the right answer will be in green text, that is option number C, the mouse local lymph node assay. So what is the explanation and the reference which is given by the American Board of Toxicology is the murine local lymph node assay measures mitogenesis of immune cells. There are immune cells are there, in that also they are specifically measuring which particular phase, mitogenesis phase only they are measuring. Okay, in the mouse lymph nodes following the stimulation. So lymph nodes will be there in the mice. After the stimulation, they are measuring only the mitogenesis phase in the lymph nodes. Okay, so LLNA assay is the only assay that measures only the induction phase of the sensitization. Other assays are there which also measure pre-sensitization, proliferation phase, all those things. So all other require a second exposure. First exposure pre-sensitization, second one is second exposure and the measurement of this elicitation phase. So there are other assays like GPMT assays are there which also measures about the elicitation phase. So along with this explanation which is given by the ABT, I would also request you also go through Hayes 5th edition. This page number we are not, I went back and checked this. There is some error with respect to page number which is given by this particular document by the ABT. But I think you can use the key terms like local lymph node assay, and if you people are having Hayes Principles of Toxicology textbook, right now though I am having 7th edition, I couldn't trace that particular this thing. So you can go back and check. So with respect to this particular question, we will also read what are the other concepts that we can learn from this particular question. So the local lymph node assay, also known as LLNA, is an in vivo test. Okay, in the mice used to predict a substance potential to cause skin sensitization. The basic objective of this LLNA assay, it is to identify the skin sensitization, and it is done in the case of mice, whereas in the case of GPMT, it is the test species is guinea pig. Then LLNA measures the proliferation of the lymphocytes in the lymph nodes. We know reticulocytes will be there, RBC, WBC, so many types of cells are there. Particularly, it is measuring lymphocytes in the lymph nodes. That drain the site of chemical application. Wherever the test chemical is applied to that particular local area, all these lymphocytes will start proliferating towards the site of exposure of that particular chemical. So that is what they say, drain the site of chemical application with the level of proliferation being proportional to the dose applied. As the dose of the particular chemical exposed increases, the amount of proliferation of these cells towards the lymphocyte, proliferation of the lymphocyte through lymph nodes to that particular exposed area keeps on increasing. So dose is directly proportional to the amount of the cells which are proliferating to that particular site. That you have to remember. Next one is LLNA method has largely replaced older. What were the older methods? Which see, whenever you are coming with a new method, no, there should be some drawback, some limitations with the old method, already existing method, which we are not able to achieve. So to overcome those limitations, always will come up with new methods. So what were the limitations of this old method? Scientifically rigorous guinea pig test. So if you look at GPMT test, you need three particular phases of exposure is needed. Okay, so the scientifically rigorous guinea pig test is there, providing a more reliable quantitative. So by using this LLNA method instead of qualitative, which is subjective, means one study director may assume that this particular reaction is erythema or redness. Another, if you ask the same animal, same reaction, if you are asking another person, another study director to observe and give a rating, he might give some other rating. Okay, so there is a subjective variation will be there. So but that is why it is called qualitative. But here in this case of LLNA, you can do quantitative estimation can be done, and it is human alternative with animal welfare benefits. So it is in line with the ethics committee also, this LLNA method. Okay, that is the reason it has been replaced. I won't say completely replaced, but it is an alternative to the existing traditional method like GPMT essay. That is what it is saying. I am not saying it. Okay, so what are the steps do we have in the LLNA? Major steps we'll see. LLNA involves applying a test chemical, whatever the chemical which you are testing. No, it can be the nature of the chemical might be drug or cosmetic or a pesticide or agrochemical, right? To the ear of the mice. So we are applying to the ear of the mice. The chemical induces a primary proliferation, that is a rapid increase of lymphocytes in the lymph nodes, draining the application site. So they will start all these lymphocytes start traveling towards the site of application through the lymph nodes, right? The proliferation is the hallmark of sensitization. So always they will ask you if there is an adverse effect or there is a certain assay. They will ask you what is the hallmark. So in this case, in the case of skin sensitization, proliferation of these lymphocytes at the site of application is the hallmark. So one question you might get that one. Okay. Then see, like this question might be proliferation of lymphocytes through lymph nodes at the site of application. You might have again four options. Like GPMT might be there. There might be Magnus and Klimet or MEST Mauser spelling test, something like that will be there. One option will be LLNA assay. Then you have to tick LLNA assay in your exam. The next LLNA assay measure this cellular responses often by measuring the incorporation of a radioisotope, because these radioisotopes, know whatever the isotope which you are measuring, if you are radio label it, you can quantify it into the DNA of the dividing cells, providing a quantitative measure of the sensitization potential. So this is how it is differing. So if you are able to if you are able to trace the isotope and radio label it, and you can quantify that particular observation. So LLNA test uses fewer animals when compared to GPMT tested traditional method. It is using comparatively more number of animals than LLNASA, and it is considered more ethical than the traditional guinea pig test. So this you have to remember. The next what are the significance and limitations with respect to this LLNA is LLNA is a quantitative method. It's not a qualitative method that provides a numerical prediction of a substance potency. So as the potency of a molecule increases, how this sensitization of that chemical. So still no what has happened, whether you take skin irritation or you can say sensitization, we were not able to quantify. So these assays were not used for risk assessment. If we are able to quantify it and have a value, now we can have one what do you say, EC50 value or RD50, or we'll know what is the Noel at which dose it is not causing sensitization. If such a numerical value, if we will able to know, no, if we are able to establish a dose response curve with respect to sensitization endpoint also, similar to that of systemic toxicity, we can use this particular endpoint for skin sensitization. There are nowadays lot of articles which are published where they are using skin sensitization as an endpoint, as a Noel to derive risk assessment. So please go back and refer those also. So a numerical prediction of a substance potency and has been successfully validated for regulatory use worldwide, particularly in the case of cosmetics, as well as it's an alternative particularly to pesticides and agrochemical, where the workers are constantly dealing with herbicides when they are applying, applicators will be there, no, for them. So what are the limitations of LLNA is it has been widely accepted gold standard. Recent guidance from the agencies like the FDA suggested that a combination of non-animal methods like in silico, in chemico, as well as in vitro can now be used to adequately predict human skin sensitization, and the LLNA may no longer be recommended for some applications. So this is the thing. Even though we are switching from GPMT to LLNA, now FDA is also recommending that please go for a battery. Similarly, like genetic toxicology studies, no, it is asking us to go for a combination of both in silico, in chemico, in vitro. Together, the results from these type of assays can be used as an alternative or replacement to the LLNSA. Always remember, see, our regulatory agencies, they want to focus towards the application more and more towards application of either 3R principle or 4R principle. So one of the key R in the case of 3R principle is always go for alternative method, reduce, refine and recycle. If there is a method that can be refined, or there is a method through which you can use less number of animals to do your study, that is reduce, reduction in the number of animals that are used for deriving a certain end point or certain conclusion. So always regulatory agencies, particularly whether it is FDA or EMA, they encourage you to replace this method with a better alternative method. So that was the thing. So I would also request you to go and read in detail OECD guideline 442B, and there is a recent revision is there in June 2025, because right now we are in the year 2025 only. So a couple of months back only there is a revision is there. So please go back and see what are the new changes are there in this particular guideline. Okay, then we will quickly go through only the comparison between the two assay, that is LLNA assay and GPMT assay. Phases measured in the case of LLNA assay is induction phase only. So our first slide, if you see, the question was which of the following assay will do only induction phase, that is LLNA assay. So in case of GPMT assay, both induction phase and elicitation phase are measured. And what is the principle with respect to LLNA is a chemical is applied to the ear of the mouse, and proliferation of the lymphocytes in the draining lymph nodes is measured. This particular LLNSA, it will quantify the initial T cell activation that occurs during the induction phase. Whereas in the case of GPMT assay, chemical is initially injected and applied to guinea pig during the induction phase, like this. Okay, then after a rest period, the animals are given a second challenge exposure. Based on the reaction, they will be given second challenge exposure to the same chemical. The extent of the skin reaction to this challenge is observed and compared to the control animals, thereby evaluating the elicitation phase. So in the case of GPMT, you have induction phase as well as you have elicitation phase. So that is the take home message one has to remember. Here very quickly some kind of diagram is given. You can pause the video and go through the principle. Principle you can read in OECD 443 guideline also. So hope from this video you were able to learn about LLN essay and GPMT essay, important difference and the application and limitations of LLN essay, how that is used nowadays for the risk assessment, right? So thank you so much. Be curious. For any questions, please write down a mail to arya.priyanka18@gmail.com. Like, subscribe and follow my YouTube channel Toxicology on Board. Thank you so much. All the best for your exam.